Okay, greetings. We're going to finish up chapter 15, the special senses. Now, what we have to first deal with is something a little bit more particular here, because what we have are cells that create what are called generator potentials, and this is a depolarization of the membrane. We're going to be dealing with 5 special senses: olfaction, the sense of smell; gustation, the sense of taste; vision; equilibrium, which is involved with the sense of balance, rotation, linear acceleration, things like that, as well as hearing. Now, when we deal with generator potentials that exist in some of the sensory cells, this is a depolarization of the membrane, which, if strong enough, can generate an action potential, an AP, in that cell or a sensory neuron. Usually you're going to have, in some cases, the sensory cell being the neuron at the same time, or it's going to be a specialized cell and it's going to be intimate, that meaning it's going to be right next to a sensory neuron. So all these special senses originate at sensory receptor cells, and we're going to explore this for a little bit. Now, the first area we're going to deal with is the sense of olfaction. Olfactory receptors are really the dendrites of specialized neurons. Receptors are responsive to selective parts of the odorant molecule. That's what I've mentioned before in labs, and I'm going to say it again. What you have to understand is that there is no receptor that is specialized for gasoline or roses or freshly baked bread. What you have are a variety of odorant receptors that attach to certain select parts of the odorant molecule. So one part might be a benzene ring, another part might be multiple benzene rings, another part might be a phenyl group, another part might be an amine group, another part might be an alcohol group. And all of this information is summated together and gives a unique odor that we record in our brain, I should say. And so what you have is something very special with olfaction. It's kind of called the synthetic method. You only see it really also with vision, because when you look at vision and you look at color vision, you have 3 receptors: red, green, blue, but they all have overlap. So that's why you can pick up colors like yellow and mauve and pink and brown and purple, okay? In this case, it summates a bunch of parts to give you the picture of what the odorant molecule is. Now, receptors for senses of taste, vision, equilibrium, and hearing, they're specialized cells that are inexcitable. They don't form action potentials. They form synapses with processes of sensory neurons. Those cells do fire off action potentials. Upon stimulus, receptor membrane generates a graded depolarization. The depolarization causes a release of a neurotransmitter to the sensory neuron. The neurotransmitter causes the neuron to trigger a generator potential, GP. The sensory neuron, GP, triggers APs in the sensory neuron to pass information to the CNS, okay? The synapse creates a synaptic delay, so there's a little bit of processing that takes some time from the original stimulus to the actual, to the AP generation. So here we have the stimulus, here we have the dendrites, specialized olfactory neuron. Notice that there's myelination here to speed the signal across. So what you have is a stimulus, and then bang, you're going to get action potential right across here. This is the generator potential that we call it. When the stimulus is removed, boom, everything's gone. When you look at this from a situation where you have stimulus and then a receptor cell, the receptor cell is going to generate these generator potentials, and there's going to be a synaptic delay because it's sending a signal over here this is the axon of the sensory neuron. So it's going to first have to get stimulated, issue some sort of depolarization. That's going to take time to pass over to the synapse. And then the axon is going to send the signals that you see right here. Now, with olfaction, that involves specialized chemoreceptive neurons and delivers sensations directly to the cerebrum. Olfaction is made possible by olfactory organs attached to cranial nerve number 1. That's the olfactory nerve. The epithelium of the olfactory organs cover the inferior surface of the cribriform plate, the superior portion of the perpendicular plate, the superior nasal conchae, and that's all ethmoid bone. The olfactory pathway consists of olfactory receptor cells, olfactory epithelium axons, that's going past the cribriform plate, the olfactory bulb, the olfactory tract, which innervates the olfactory cortex, hypothalamus, and part of the limbic system. In other words, what I'm saying to you is that this very, very primitive, this very, very old style chemical detection system passes over to areas of emotion, the hypothalamus, to the olfactory cortex, which of course processes some of the information, part of the limbic system, and that's going to be also emotion. In other words, let's face it, we have similar reactions between a rose and a baby's dirty diaper, right? And you can picture it. You have that emotional response, okay? Now, I want you to be aware of a couple of other things. What's anosmia? Anosmia is the loss of the sense of smell, either total or partial. It may be caused by head injury, infection, cadmium poisoning, or a blockage of the nose, such as a nasal polyp. When we talk about the organ composition of olfaction, the olfactory epithelium consists of knob and dendrites. These are the receptors are present on the dendrites. The supportive cells, the basal cells, these are regenerative stem cells, and they replace worn-out olfactory cells. Roughly, the olfactory cells have a lifespan of about maybe 10 days or so, and they're constantly being turned over. You also have a lamina propria, and we have olfactory glands, which basically secrete a mucus-type material. It's mucus and water, and it helps keep the site moist. Odorants are the terms used for dissolved chemicals that stimulate the olfactory neurons. Generally, they are small, airborne, lipid-soluble, and water-soluble molecules. So something that is neither lipid-soluble or water-soluble may not be detected by olfaction. Odorants are odorant binding proteins. These are located on the olfactory dendrites. An odorant receptor will activate a G protein process inside the olfactory receptor cell. And you can see this over here. Here we have this process. Olfactory receptor cells, bundled axons penetrating the cribriform plate, going right through there. And this is all ethmoid bone. This information comes up to the bulb. The bulb processes the information, passes it through the tract, and this comes up into the limbic system and hypothalamus, as well as, of course, the olfactory cortex. Now, here you see the olfactory gland. That's going to continue to keep this area kind of moist. You have the olfactory dendrites. These, on their surfaces, contain the receptor proteins. At the end of them is sort of this area of fusing of all the dendrites, which is called the knob. You have the supportive cells here, and here. And what you have here are also basal cells that will divide and replace worn-out olfactory cells, okay? Notice that the cell has an axon going directly upwards, superior into the olfactory bulb, okay? And just a couple of things to keep in mind, and that is the receptor cell. The receptor protein, you have an odorant molecule attached to it. This activates a G protein complex, which makes cyclic AMP. The cyclic AMP will open up a closed sodium channel. The sodium, which is ions which are on the outside of the membrane, come in, rushing in, causing a depolarization. If there is sufficient depolarization, the action potential is triggered in the axon, and the information is relayed to the CNS. Next we're going to go into is gustation. That involves epithelial chemoreceptor cells located in the taste buds. Gustation is the sense of taste. These receptors are located on the superior surface of the tongue, but there are receptors, taste receptors, that exist on the surfaces of the pharynx, larynx, and epiglottis. Now on the tongue, there are 4 types of lingual papillae. VALLATE, which in some books we'll call it circumvallate, foliate, fungiform, and filiform. Now, just keep this in mind. VALLATE contains up to about 100 taste receptors. Foliate has some. Some different texts will say 10 or so, but it depends. Fungiform has about 5 taste receptors. Now, the interesting thing is that filiform really is more to provide friction, to move food around. You've got to have some adhesion or some type of contact between the food and the tongue. There are no taste buds. The primary taste sensations are sweet, sour, salty, bitter, and then umami, which is the meaty, savory, brothy sensation discovered by the Japanese in the early 1960s. The receptors are sensitive to amino acids, small peptides, and nucleotides. Here you see the VALLATE papillae. There is umami detected around here. We have sour, bitter, salty, sweet. And you can see up close the VALLATE papillae, the foliate, the fungiform, and then the filiform. Now, notice over here where it says water receptors, pharynx. Okay, let's go over that. We found that there are water receptors that exist in the pharynx, and they provide information to the hypothalamus upon drinking of water. Now, this is important because it's always interesting to find new areas, and what these are just cells that will respond to the presence of water and signal back up through the pathways to the hypothalamus. Okay. When you look at a primary receptor cell for gustation, they have these microvilli. They refer to them as taste hairs, which extend into the surrounding fluid for chemical sensations. Interestingly enough, a typical gustatory taste cell lasts about 10 days also. There are basal cells which will produce transitional cells which eventually develop into replacement gustatory receptor cells. Now there's an interesting comparison here. The sensitivity to taste. The taste receptors, they found that they're more sensitive to bitter, which by 100,000 times, or to acids, which by 1,000 times, than to sweet or to salty. There is a value in this, in that it prevents the consumption of toxins. Now, if you think about this, even if in a basic wilderness environment, our great ancestors or whatever, you probably have heard of plant alkaloids. A lot of plants that have toxins, these toxins come up alkaloid or bitter. In acids, you're always going to get that sour taste in something. Sweet or salty is a little bit different. Salty is merely just detection of like sodium ions. You may get some sensitivity also to potassium ions and lithium ions and things like that. But there is a definite advantage for survival in preventing consumption of toxins. Now, the interesting thing also is that as we age, the taste buds decline in number after age 50. And in a lot of cases, it's best demonstrated. Elderly do not have strong sensitivity to taste as the young. Okay, so here's our transitionary cell. This is basically a diagrammic view of a taste bud. You have the basal cells here just setting up shop. Transitional cells will eventually replace these functional gustatory receptor cells, and their cells have these taste hairs, which are the microvilli, which really have the detection of the different forms of taste. Now, what is it that we're actually tasting? First off, you have to understand that gustatory reception relies on membrane receptors and ion channels, and sensations are carried by the facial nerve, which is cranial nerve number 7, the glossopharyngeal cranial nerve, which is cranial nerve number 9, and the vagus nerve, which is cranial nerve number 10. Gustatory receptors are stimulated by dissolved chemicals. You can't dissolve in water or fluid, you really aren't going to be easy to be detected. Dissolved chemicals stimulate hair cell membranes. Here's what's going on. If they diffuse through the plasma membrane, leak channels, that is sodium leak channels, sodium ions, for the salty and hydrogen for the sour chemicals will basically be able to detect those chemicals. Membrane receptors that activate their G proteins are called gustaductins, and they are protein complexes that use secondary messengers to stimulate the cell effects, which is the neurotransmitter release. This you will find for the tastes of sweet, bitter, and umami. And you can see this right here. So salt and sour. Salt will obviously be predominantly sodium. Sour is something with hydrogen ions. They leak through and they're going to cause a depolarization. Sweet, bitter, umami, you're going to have specific membrane receptors that will attach to these. The different receptors are specific for compounds that are sweet, compounds that may be bitter, or umami, which we've mentioned before. Now let's talk about the neural pathways. All basically input from the facial, glossopharyngeal, and vagus nerves, and all of this information is passed to the solitary nucleus. Remember that? That's the one that's in the medulla oblongata. The axons of the postsynaptic neurons cross over and enter the medial lemniscus of the thalamus. After another synapse in the thalamus, the final axon is projected to the gustatory cortex of the insula. Now, the level of olfactory stimulation plays a key role in taste perception. Smell enhances taste sensory perception. What I'm saying there is, and I think I mentioned this, is if you had your nose completely plugged off, you would not be able to really enjoy your food. You might be able to detect the tastes of bitter or salty or sweet, but not the full taste and the aroma and all the wonders of a baked potato or a steak or spaghetti and meatballs or anything else. Okay? Anyways, let's move on. And you can see these neural pathways that come up, CN10, 9, 7, all coming up to the solitary nucleus of the medulla oblongata, which then have a crossing over on the medial lemniscus of the medulla oblongata and eventually going over to the gustatory cortex, which is then passing over to here, the insula. So over here you have the thalamus, and the information is then passed over to the gustatory cortex. The next area we're going to deal with is vision. Now, the eyes form early in embryonic development. At 4 weeks, we have eye development starting as optic vesicles, or bulges, on the lateral walls of the prosencephalon. By 5 weeks, though, the optic vesicles become indented. They form a pair of optic cups that remain connected to the diencephalon by thin stalks. The epidermis overlying the optic cups begins to develop into a pocket that pinches off and becomes the lens of the eye. This is where we start getting into specialized layers of retinal development, because at that point, ependymal cells of the outer layer of the optic cup develop into photoreceptors. Ependymal cells of the inner layer of the optic cup develop into pigmented cells layer. The nervous tissue of the outer layer of the optic cup forms neurons, ganglion cells, and the specialized glial cells of the upper layers of the retina. This is for processing the light information. And by the time we get to the 6th week, optic nerve CN2 follows the path of the original stalk connected to the optic cup. Embryonic cells are surrounding the optic cup. They will form supportive layers of connective tissue to isolate the nervous tissue from the rest of the body. The eye is going to develop interior chambers. They are filled with fluid that is actively made and reabsorbed. So we go from week 4, optic vesicle, neural tube. Week 5, the developmental structures of the retina, including the optic cup and the developing lenses here. And eventually week 6, where you can clearly see now you have eyelids, fluid-filled chambers, a developed lens, connective tissue that has now become capable of sequestering and isolating all that neural tissue there. Next, let's deal with a fully constructed eye. We're going to start off with accessory structures of the eye that provide protection while allowing light to reach the interior. Keep in mind there's some very important points around this. You don't want light echoes or abnormal areas of light to come into the eye, which could lead to confusion of whether you're looking at the image or some other fused or botched or mixed up images. When we look at the accessory eye structures, we have to first consider the cornea, which is the transparent area of the interior surface of the eye. Then we have eyebrows. That's thick hair bands that basically protect the eye from perspiration and direct sunlight, whereas the eyelashes are thick eyelid hairs that protect the eyes from particulate matter. Eyelids, this is referred to as also palpebrae. These are fleshy eye coverings. Blinking the eyelids will moisten the eyes. Also, the eyelids contain modified sebaceous glands. This is called the meibomian glands. And basically, the secretions prevent eyelids from sticking together. We also have to consider the conjunctiva. These are mucous membranes that line the inner surface of the eyelid. You have the palpebral conjunctiva, which is the inner surface of the eyelid, and the ocular conjunctiva, which is the interior surface of the eye. This is interesting because, as I mentioned in class before, it's the conjunctiva that really keeps the contact lenses. People will say, my contact lens, it's going to go back to the back part of my eye. Can't do it. Can't do it. As long as the conjunctiva is there, it's going to keep it sequestered in that general area of the eyelid thing. Not going to go anywhere. You have the palpebral fissure, and this is the gap that separates the free margins of the upper and lower eyelid. Now we deal with the canthus, otherwise known as, the lateral angle of the eye, which would be the lateral canthus. That's the lateral side that the two eyelids are connected. And the medial canthus, which is otherwise known as the medial angle of the eye, and that's where the two eyelids are connected on the medial side. We have a lacrimal caruncle, which is a reddish mass of at the medial canthus. The glands produce mucus while sleeping. That's that crud that you wake up to when it's kind of in the corner of your eyes and such. We also have the pupil, which is the opening at the center of the iris where the light enters the eye. Now, here's the pupil. Okay, we're going to get more into what the iris is and etc. But this is the cornea. Notice there are no blood vessels. It is very clear. We have the lateral angle of the eye. We have the lacrimal caruncle. We have the medial angle of the eye. We have the palpebrae fissure, the eyelid, and the eyelashes. And of course, up here would be the eyebrow. When we look at this, we also have several other structures to consider. From a side view here, you have, of course, the corneal epithelium. You have also this fornix, which is really the corner where you have a fold here of the palpebral conjunctiva and the bulbar or ocular conjunctiva. Okay? And as we move on, we have to keep in mind that tears are very important. Tears contain lysozyme and antibodies that are going to kill most bacteria. When we look at the tear system, the lacrimal apparatus, we have, of course, the lacrimal gland, otherwise known as the tear gland. This is located inside of the superolateral margin of the orbital cavity. From there are lacrimal ducts that roughly in the range of 10 to 12, and they transport the tears to the upper eyelid. The lacrimal puncta, which is also referred to as the lake of tears, that's the drainage pores located in the lacrimal caruncle. The lacrimal caniculi, these are small canals that connect the lacrimal puncta to the lacrimal sac. The lacrimal sac is a drainage from the lacrimal puncta, and then you deal with the nasal lacrimal or otherwise, duct, otherwise known as the nasal lacrimal duct. This drains tears from the lacrimal sac to the nasal cavity. Notice that the layer of orbital fat posterior to the eyeball is providing padding while allowing the eye movements. Now I bring this up because of the fact that when you look here, here is your lacrimal gland. The fluid moves from lacrimal gland through the ducts over the eyeball, all right? Comes to the lacrimal puncta, goes in through the lacrimal canal into the lacrimal sac, and eventually empties into the nasolacrimal duct, which it leads into the inferior meatus. If anyone has ever done some serious crying, you know that after a bit you've got to sniff, sniff, sniff. You've got more mucus and really tears that are in the nasal area. And it's because of this, how can I say it, this basically passageways that lead to the loss of tears from the eye going into the nasal area. Notice the comment, as I mentioned before, the orbital fat here and here. Now, excuse me, the hollow eyeball has a layered wall and a fluid-filled anterior and posterior cavities. First off, keep in mind also that the eye is supplied by optic nerve number 2, CN2. There are 3 tunics or layers of the eye. First, the fibrous tunic. The sclera is the tough opaque membrane, and it contains collagen elastic fibers and protects the shape of the eyeball. It also prevents light from coming in other directions other than through the pupil. The cornea, which is transparent to allow light into the eye, lacks blood vessels. The border between the sclera and the corneoscleral junction, otherwise known as the canal of Schlemm, it's also known as the scleral venous sinus. This absorbs the aqueous humor. We're going to get into the aqueous humor and how it's moved around in a minute. The functions of the fibrous tunic include supporting and protecting the eye, attachment site for extrinsic muscles, as well as containing the transparent cornea. From the fibrous tunic, we move to the vascular tunic, which is basically choroid, contains numerous blood vessels and melanocytes to produce melanin to block light reflection out of the eyes. So basically, you have to have a very active, functional blood vessel system in the eye to provide the nutrients necessary to keep the neural tunic alive, but also you've got to keep a certain amount of melanocytes present to just keep from light coming in or leaving from the eyes. Now also, the other functions for the vascular tunic include basically providing a route for blood vessels and lymphatics to supply the eye tissues, as I mentioned before, regulate the amount of light entering the eye, the secretion and reabsorption of the aqueous humor, and control the shape of the lens. This is part of the focusing process. The anterior extensions to the vascular tunic include the iris and the ciliary body. The iris controls the light entry into the eye and hence controls the size of the pupil. The ciliary body is a ring of smooth muscle that adjusts the shape of the lens for focus. We're going to get into the lens and how that works in a few minutes. Then we get into the neural tunic. This is the innermost layer consisting of the retina, which contains both rods and cones. Now, you get confused. Which one does what? Rods are responsive for light. Cones, color. Just think of cones and color. You have 3 types of cones: red, green, blue. We'll get into that. Basically, the neural tunic consists of a thin outer layer. This is the pigmented layer to absorb light, and a thick inner layer, innermost layer, which is the neural layer that contains the photoreceptors. And you can see this here, fibrous layer, sclera, cornea. And then we're going to have the vascular layer, which is going to include the iris, the ciliary body, and of course the choroid. And then the innermost layer, which is the retina. Now, when you look at the eye from a cross section, you're going to notice that there are cavities of the eye. We have these anterior cavity and the posterior cavity. We've got to deal with the anterior first because it's really subdivided into the anterior chamber, which extends from the cornea to the iris. Now, the posterior chamber extends from behind the iris to the ciliary body and the lens. They're filled with this very thin, transparent fluid called aqueous fluid. And the flow and the production of the aqueous humor goes from the ciliary body through the ciliary processes and the ciliary zonial through and going over the lens, and we'll get into this in a second. It's going to go out into more the anterior chamber and eventually to be absorbed by the canal or Schlemm. So what we have here is the production of the aqueous humor being produced through here, moving up through and around and eventually absorbed here. This is the ciliary body. These are the zonials. These structures actually attach to the lens and will pull on it. And as they do, they either flatten the lens or they allow the lens to become much more curvy to focus the image on the back part of the eye. Well, what about the posterior cavity? It's filled with a gel-like material called vitreous humor. And the circulation of the humors is critical for the nutrient and waste transport. I almost forgot a point, and this is that the aqueous humor really provides nutrients, and internally, on the internal side of the cornea, gas exchange can occur because there's no blood vessels in the cornea, and they're never supposed to be. If it was, then you'd have a real serious problem in being able to see through that cornea. Okay? Now, as I said before, here's the movement. The ciliary body produces right around in here. These are the ciliary processes. This is the ciliary muscle. These are the ciliary zonule. The suspensory ligament is another way that they have referred to them. And here is this wonderful lens made of very transparent proteins called crystallins. This is the aperture that light will go through the pupil. The aperture is controlled by the iris, but as the fluid moves around underneath the lens and goes out, it will be eventually absorbed by the sclerovenous sinus here. Okay. I've mentioned what the cornea is. I've mentioned what the sclera is. The iris is colored, and its opening controls the light entry through the pupil only. I've mentioned what the lens is. You would also be mindful to understand that the aqueous humor and vitreous humor are transparent. The macula is the site of the highest concentration of photoreceptors. Now we're going to move to the back part of the eye. We have a section called the fovea centralis. It is a shallow depression. The central area of the macula is the point of focus. You see it back here. And what happens here, interestingly enough, in the fovea itself is that it is loaded, basically filled with cones. That is the site of the greatest image sharpness. I bring your attention also to optic nerve number 2 here, but also notice this little site here. This is known as the blind spot because here is where you have all the axons of the ganglionic cells that are part of the retina coming down through and heading out toward the brain. Also, what pops out are the central artery and vein, so there's no receptors here. This is a spot where there is no vision or image that is picked up. Now, interestingly enough, the visual axis is an imaginary line for image pathways from the center of the object image through the cornea center and to the center of the retina. The control of the light entry, as I said before, was by the iris. And what interestingly enough is that the pupillary size is controlled by two different smooth muscles in the iris, the dilator pupillae, the sphincter pupillae. Now, the dilator pupillae extends radially away from the center of the edge of the pupil, and contraction will actually enlarge the pupil. It's activated by sympathetic stimulation. The sphincter pupillae forms a series of concentric circles around the pupil. Now, this is interesting because when the sphincter itself contracts, the pupil diameter decreases. This is activated by parasympathetic stimulation. So here you have it, dilator pupillae, okay? The dilator pupillae is out here. It's like spokes on a wheel. And so when they begin to contract, they pull out. And so the pupil increases. This is when you see this as, for example, in increased sympathetic stimulation. Here, this little ring here, the sphincter pupillae, another set of muscles, all smooth muscles, by the way, when it is activated, will contract, and when it contracts, the pupil gets much smaller. This is increased by sympathetic stimulation. We're going to see how this is important because it is really key to controlling whether we're going to have fine, close-up, detailed vision or broad field, but not as sharp, wide field vision. Now, when we focus, the focusing of light produces a sharp image on the retina. The light is refracted, that is bent, when it passes from one medium to another. For example, if you've ever put in a spoon or something into a clear glass of water, you notice that the image seems to shift when you go from one medium to another, from air to that water. The same thing occurs when you go from air to cornea, but it's not as much with the cornea, but the lens. The lens will cause a major refraction that occurs, okay? And this will play a key role in focusing the light rays from the object towards a focal point on the retina. A specific point of intersection of light rays on the retina is referred to as a focal point. The distance between the center of the lens and the focal point is referred to as focal distance. Now, the term accommodation, this is a process of focusing an image on the retina via changing the shape of the lens to keep the focal distance constant and provide clear vision. You do it all the time. You do it all the time, you don't even recognize it. If you move your visual field from, let's say, 10 feet away to 30 feet away, you're supposed to be able to have accommodation. So the lens curvature is controlled by these ciliary muscles pulling on the suspensory ligaments to change the shape of the lens. For close vision, the ciliary muscle contracts and the lens is rounded. For distant vision, the ciliary muscles relax, the lens kind of flattens out, kind of relaxes. Now, the greatest amount of refraction is required to view objects that are very close to the lens. Near the point of vision, this is the inner limit of clear vision for close objects. This is determined by the degree of elasticity of the lens. Now, the elasticity of the lens declines as we age, as well as the effectiveness of the suspensory ligaments to the lens. This results in increases in near point of vision after age 40. In other words, That's when you get reading glasses. Now let me also bring up one other point. If you go to the image right here, or you can take it right here, what happens also, interesting enough, is if you can see the diagram here and here, the suspensory ligaments are right on the edge of the lens. Well, it's been found by medical studies is that they start migrating downward to the lower part here, or to the posterior part of where the lens is. They don't go all the way down to the bottom, but they stay lower. And this means that as the ligaments pull, excuse me, they're not going to get the same rounding that you would get required for near vision. This is one of the other reasons why we need, as we get older, reading glasses, because we're just not able to have that magnification for up-close imagery. Now let's go into the neural layer of the retina. It contains multiple layers of specialized photoreceptors, neurons, and supporting cells. We have a pigmented layer. Light passes photoreceptors and stops at the layer to prevent light reflections, and what's referred to as visual echoes to the retina. If you remember when you did the dissection of the cow eye, you notice that sort of iridescent backing of the eye. That's the tapetum lucidum. Now that happens with certain animals. It doesn't happen with people. Instead of light reflecting off the back wall and then coming back to activate more receptors in the retina, we don't have that. It passes through the rods and cones, and if it doesn't activate it, boom, it crashes and gets absorbed by the melanin in the melanocytes in that pigmented layer. The neural layer contains rods which provide vision in black and white, high sensitivity to light. So in other words, it's great for dim light, bright light, etc. The cones provide color vision, red, blue, green, but they have overlap. We'll talk about that in a second. The key point that you need to understand, though, is that cones require more intense light for stimulation. That's why I may have mentioned this in class, that if you look down on something and it's at very low light, if you actually look slightly off from center, you'll move the image into an area where there's more rods, less cones, and you may be able to see it better in this low light. Now, if you look straight at it, you're basically pushing the image onto an area that is basically fully just cones. They need more light. After the cones and rods, you have that signal processed through another set of cells called bipolar cells. These are photoreceptor cells that synapse with these cells. So the rods and cones will synapse with them, the bipolar cells. The bipolar cells will synapse with ganglionic cells. From these ganglionic cells, axons of these cells leave the eye and become the optic nerve, as you can see here. So you have rods and cones, bipolar cells, ganglionic cells, and their axons join together and leave. This area I mentioned before, the optic disc, if you notice, there's no rods, no cones, only axons and blood vessels. That is, the optic disc is basically the blind spot in your visual field. If you notice this is an up-close image, an excellent image of the back of the eye. Here is the macula. In the center of that, ideally, is where you would want your images, for the most part, viewed upon, because that area is the sharpest for image processing, and it's the fovea centralis. Okay? Over here is the optic disc. This is where all of the axons from all over the visual field are going back down in, and that's going to form the optic nerve. Also, you notice the blood vessels coming out and going in. Okay? Now, there are a couple of horizontal layers that are with special cells that process and synapse to do some processing of the image and the photons that have activated the different rods and cones. The horizontal cells are the first layer. They alter sensitivity of the retina by synapsing between the photoreceptors and the bipolar cells. The next layer is the amacrine cells. They alter sensitivity of the retina by synapsing between bipolar cells and ganglionic cells. Both play a role in the eye's adjustment to dim and brightly lit environments. I've mentioned what the blind spot is and what the visual acuity. The greatest density is in the cones area, where it's all cones. And you can see where, first off, the horizontal cells exist right here between the rods and cones and the bipolar cells. The amacrine cells are here. They synapse in between the bipolar cells and the ganglionic cells, okay? And you notice the ganglionic cells, they have these very long axons that then go and form the optic nerve. Now, here's another thing. This is the density of cones and density of rods. You notice away from the image, what do you have? A higher density of rods. The image would be, let's say, right here. Sounds good. In sunlight, you would have no problem with foveae centralis. You've got this extremely high density of cones there. Great. Sun goes down. It's twilight. You don't have as much light available. You're going to see less and less and less. The way to take care of that image is to move the image over. Look at it askance. Look at it at the side. You'll start hitting higher density of rods and you'll be able to see and recognize the object. Can't see color, but that's okay. Now the photoreception occurs in the outer segment of the rods and cones. This is where we get into what the rods and cones are made up of and how we go through the biochemistry of vision. First off, both rods and cones have two segments, an outer segment and an inner segment. The outer segment contains flattened plates that are also referred to as discs, which contain the visual pigments. The inner segment of this cell contains the majority of the organelles, and it's responsible for most of the cell's functions, okay, biochemical, etc. The visual pigments are rhodopsin components found in the rods. You can see that basically the same ones in the cones too, but we'll get into that in a second. Rhodopsin is composed of opsin, which is a protein, and retinal, which is a derivative from vitamin A. Someone with a vitamin A deficiency will have difficulty in being able to have normal vision. Some cases with extreme vitamin A deficiency, they will become blind. But in other cases, they just become what is referred to as having night vision, or they can't see well in very low light, okay? Retinal is the pigment molecule. But let's get into this a little bit further. We see this in rods. The only difference between rods and cones in some ways is that cones have variations of the opsin that react at different wavelengths, that is, different colors of the light. The rods, remember, are reactive to both dim and very high levels of light. The cones have to have a higher level of light, but then they specialize in certain frequencies of light, certain colors, okay? Now, the color detection. If I got just red, blue, and green, do I see the universe in just red, blue, and green? Of course not. What about orange? What about yellow? Oh, what about that wild purple color? Or what about brown? What about pink? Well, all of this is due to the wavelength overlap, which I'll show you in the next diagram here, but let me go through this. Here we have the inner segment, the outer segment. The outer segment, interestingly enough, look, notice light is going this way. These guys are pointed in the wrong direction. No, they're not. This is a cone. See how it forms a cone shape? Each one of these little lines designates a disc. Those discs contain the rhodopsin light reactive molecule. Rods have cones as well, have discs as well, and they are much more straighter, but they're also much more reactive to different intensities of light. Right next to them is the backstop, as I call it. That's this heavily pigmented area from the melanocytes that basically stop light from bouncing back and creating false echoes of light. Okay? But light has to pass all the way through the retina to activate these discs. Now, cones, notice that they start and get activated in the deep violet, but peak in the blue, but then can straddle all the way into almost the yellow. Green comes out to about the blue range and is reactive all the way out to about the orange, and red goes into the intensity of the red color, but yet it can also be activated to a lesser degree all the way out to the bluish green level. These overlap in such a way to help your vision be able to detect the variety of colors in the visual spectrum. Okay? Now, how about the actual photo reaction between photons of light, in other words, the actual packets of light, and your retina? What's going on? The photoreception will involve activation, bleaching, and reassembly of the visual pigments. Now, at the resting state, there are ions moving into the outer membrane via these gated sodium ion channels. This is all due to the presence of a unique molecule called cyclic guanosine monophosphate. You've heard of cyclic AMP. Well, this is a simple relative of that. It's called cyclic guanosine monophosphate, or cyclic GMP. Now, this activity affects the outer segment and is referred to as the dark current. So it's constantly just kind of going on there in a resting state. You have a minus 40 millivolt charge on the membrane, or voltage on the membrane. What happens is that you have a rhodopsin molecule. And remember, rhodopsin is present in all of these discs that are held inside of these outer segment membranes, okay? Kind of like just stacked up there. And the rhodopsin molecule has a retinal part that can exist in two states. A linear state, it's called 11 trans, and a bent state, 11 cis. Now, upon activation by light, the retinal shifts from 11 cis to 11 trans. The shift activates the opsin molecule, and the opsin molecule then activates transducin, a G protein bound to the disc membrane, which by its way, it's transducin, Transducin, not the G, transduction, drucin, it's transducin. This is a G protein bound to the disc membrane, which in turn activates an enzyme. This enzyme, by the way, is called phosphodiesterase, PDE. Now, what does PDE do? We'll talk to you about that in a second. Here are our discs. Here is the resting state. Sodium is coming in, but it also gets kicked out. Once I get hit with light, the molecule changes. This is the retinal here. Here is the retinal here, but it's changed shape. It goes from 11 cis, boom, to 11 trans. This is because light has hit it, absorbed it. It flips the state. This causes the opsin then to act. The opsin activates transducin, which then activates PDE. PDE breaks down cyclic GMP. Remember, cyclic GMP, as long as it's here, sodium channels are going to keep allowing sodium to come in and out, in and out. No more cyclic GMP, sodium channels are inactivated. What happens now is there's no sodium coming in. There's sodium still going out. And so you end up hyperpolarizing this entire membrane. And you decrease the neurotransmitter release to adjacent bipolar cells. It's sort of a reverse thinking. Well, wait a minute. If I activate this cell, I get neurotransmitter out here. Yes, but if I don't activate it, and it's normally been activated, see right over here, neurotransmitter release. It's the absence of the release that activates the cells. As I said, PDE breaks down cyclic GMP levels, blocks the sodium ion intake. The sodium ion level decreases, it reduces the dark current. The membrane drops to minus 70 millivolts. Dark current is basically minus 40 millivolts. You've now gone to a hyperpolarization. That causes a reduction in neurotransmitter release. This is detected by the bipolar cell as an absorption of the light photon. Now, one more thing. What do I do once I've already had this flipped to an 11 trans? Well, the rhodopsin molecule can't respond to additional photons until the opsin is returned to the 11 cis shape. So what happens is all the rhodopsin molecules have to be used up. They are then broken down into retinal and opsin in this process that's called bleaching. In other words, one molecule of rhodopsin flipping isn't going to do it. When you have everything done, then you flip, the rhodopsins break down, you have this bleaching occurring. Once the bleaching occurs, opsins can be converted from 11 into 11 cis, from 11 trans. How does that occur? By ATP. Once you have a normal opsin functional, a normal retinal functional, okay, then opsin and retinal are then reassembled to function rhodopsin. I want to make sure I said that right. Here's opsin, here's the retinal. Retinal is at 11 cis. We got to flip it back to 11 trans. Did I say that 11 trans? Yeah. I had 11 trans, excuse me. I've got to go to 11 cis. I apologize for the inconvenience here. This is going to require ATP. ATP will go through this with an enzyme. You'll lose a phosphate, but as a result, you're going to change the structure of the molecule. No longer straight, bent, then you have easy assembly. And then you're back to the original state. Now, with vision done, we're not yet done. What? We got to talk about the pathways for visual information to go from the eye to both cerebral hemispheres. We got to also talk about one other thing, and that's called depth perception, the ability to judge depth or distance by interpreting the three-dimensional relationship among objects in view. This is kind of important. You have to think about this. Now, what's going on? Well, I've got two eyes, and they're set in front of me. So I have binocular vision, which means I should have stereoscopic vision. The visual pathway consists of the following. Retinal photoreceptors, information passed to optic nerve, CN2. Information then passed to optic chiasm. Information passed optic tracts to the lateral geniculate bodies. This is part of the thalamus. And then you have it sent out to projection fibers. This is referred to as optic radiation. This is basically just bundles of projection fibers that will link the lateral geniculate body to the visual cortex. So basically from the thalamus, it heads out to the visual cortex. There's visual cortex of the cerebral hemispheres, then you've got visual processing. If you remember, you're going to have first the primary visual processing and then the association visual processing. You got to keep in mind, though, that each eye receives a slightly different visual image, and this is due to A, the fovea centralis are basically 5 to 7.5 centimeters apart. B, the nose and the eye sockets block a portion of the view. And one other thing. Now, some visual information is passed to the superior colliculi. This is part of our visual reflexes. Some of the visual information will also be passed to the diencephalon and to the brainstem. This will be also necessary for survival in one sense or another. And you can see this here. So you're going to have your visual fields, part of it on this side, part of that on that side, and part of the signal actually gets basically split by the optic chiasm. How? Everything on this side and everything on this side will get passed down on the same optic track. It will go through the lateral geniculate body, There will be projection fibers. This is the optic radiation, and all of these fibers will pass the information over to the visual cortex on this side. All the visual field information on this side and on this side comes together, passes down this pathway. By the way, note that there are some pathways, collaterals of optic fibers to the diencephalon and brainstem, as well as to the superior colliculi. Okay? Here's another thought. What happens when you have like nearsightedness and all that? How does that occur? Refractive problems result from abnormalities in the cornea or lens or in the shape of the eye. Now, if you have normal vision, it's called emetropia. That's when the ciliary muscle is relaxed, the lens is flattened, and the image of the distant object is focused on the retinal surface. Everything is fine. You're in focus. Ever notice that you have those vision charts, the Snelling charts, and they say you got to be 20 feet away. If you have 20/20 vision, you'll be able to see that line that's marked 20/20. That's the distance. Now, what is your nearsightedness? This is usually due to the eyeball is too deep or resting curvature of the eye is too great, and near objects are clear, but distant objects are out of focus. That's called myopia. Hyperopia is farsightedness. This is due to the eyeball being too shallow or the lens is too flat. The ciliary muscle must contract on distant objects, and at close distance, the lens cannot provide enough refraction to focus the image on the retina. Now, of course, all of these situations can be corrected by external lenses, usually glasses or contacts. As you can see here, here's myopia. Because of this, the focus of the image is really early before it's supposed to come in contact with the retina. If you correct this with a concave lens, you can move the image over to being in proper orientation with the retina here. Same thing with farsightedness. Notice that in farsightedness, the actual focus of the image is beyond the range of the retina. With corrective lenses, a convex lens, you can move the image focus from the basically beyond the depth of the eyeball back onto the retinal surface. The next area we're going to deal with is going to be equilibrium and hearing. Now, both of these senses involve the inner ear. Hearing is cochlea. The equilibrium is from the semicircular ducts and the saccule and utricle. Now, the sensory receptors are located in the inner ear where the information is processed prior to delivery to the CNS. We need to get familiarized with a particular structure that's important in this, and that is the sensory receptor called the hair cells. These are structures that extend outside the cell membrane, and they're similar to cilia or microvilli. Hair cells have these unique structures that come out, and what happens is they're basically mechanoreceptors. They basically transmit neurotransmitters upon forces of displacement put against the hairs. So usually what you're going to have is the hair cell having hairs coming out of it, and right next to the hair cell it is going to be surrounded by a sensory neuron. The transmission of the neurotransmitter from hair cell to the sensory neuron initiates AP signal, or action potentials. Now the cilia, the hair, may be referred to as stereocilia, as they resemble very long microvilli. The kinocilium is a unique situation where you have one very, very long, large cilium. It's found in the sense of balance hair cells. As you can see here, we focus in, we're getting into where the cochlear is, as well as semicircular canals, etc. In this case of the hair cell, note that it is surrounded by the dendrite of a sensory neuron. We have the stereocilia here, and in this case, we have a kinocilium, which is an extension here. It's usually, this is the leader, and providing displacement in the direction stimulates the hair cell. So you can push them this way, push them that way, etc. We're going to have several structures show you how these modified structures have the cilia or the kinocilium, stereocilia, embedded into them. Now, first off, let's deal with the ear. The ear is divided into the external ear, the middle ear, and the inner ear. On the external ear, we have the auricle, otherwise known as pinna. We have, of course, the helix, which is the cartilaginous rim, the lobule, which is referred to as the earlobe. That's the fleshy part, and the external acoustic meatus. And we also have the tympanic membrane, otherwise known as the eardrum. Beyond the tympanic membrane, we have the middle ear, which is an air-filled chamber lined with mucous membrane. Attached to the tympanic membrane are the auditory ossicles. There's 3 of them. So you have one set on one side and one set on the other. We have the malleus, otherwise known as hammer. It's attached to the tympanic membrane. We have the incus, otherwise known as the anvil, and this is attached in part to the malleus as well as to the stapes, and it transfers vibrations from the malleus to the stapes. The hammer takes the vibrations from the tympanic membrane. The stapes, otherwise known as the stirrup, transfers the vibrations to the oval window of the inner ear, and this is the cochlea part. Now, one other interesting point to note is that you have the pharyngotympanic tube, otherwise known as auditory tube or eustachian tube. This allows equalizing of air pressure from the nasopharynx to the middle ear. And you can see this right here. So here we have the external ear, the external acoustic meatus, we have the tympanic membrane, and attached to it we have the hammer, and we have the anvil, and then we have the stapes. Okay? We have the auditory tube. This allows equalization of pressure on this side with basically outside. If you had low pressure here to the point, such as if you were in an airplane, you're traveling at 32,000 feet, the pressurized cabin is only good and basically provides for about the equivalent of an elevation of 8,000 feet. The pressure is lower out here than it is in here, and so what happens is the tympanic membrane tends to bulge outward until you repressurize this. Yawn, sometimes eating helps on a flight, etc. Note also that you're going to have several other important structures here. Here is, of course, the cochlea, which will process sound information, and these structures here, the semicircular canals. Notice also that you have the vestibulocochlear nerve, CN8. It will take and branch off into a vestibular nerve as well as to a cochlear nerve. We need to note also that in the middle ear, there are 2 muscles. They protect the hearing, interestingly enough. And we have the stapedius, which is innervated by cranial nerve number 7, and it basically pulls on the stapes to help reduce sound damage, intensity of sound. The tensor tympani is innervated by cranial nerve number 5. It pulls on the malleus and stiffens the tympanic membrane to reduce sound damage. The cochlea, which is in the inner ear, is a snail-shaped structure containing the sensory receptors for hearing called the organ of Corti. You want to note that the cranial nerve number 8, the vestibulocochlear nerves, branches into vestibular and cochlear nerves, as I mentioned before. Basically, they're going to innervate the respective organs, the vestibule or the cochlea. The process of hearing is referred to as an example of transduction. What I mean by that is you're converting sound wave energy into mechanical energy, and this is through the tympanic membrane, the ossicles. Eventually, you transfer it into fluid waves in the cochlea, and eventually from there into neural action potentials, APs. Here is an example. What you see here is the stapedius muscle right here, which will grip on, which has an attachment to the anvil. You have also the tensor tympanic membrane, tensor tympani muscle here, which is attached to the malleus. Okay, malleus, incus, stapes. When these tense up, this reduces the sound intensity. And this can happen when you have such situations. You're in a loud area. This is going to help protect the hearing, but it does not work with some vastly sudden sounds. For example, a gunshot, a lightning strike, or something else like that, which is really brief and you can't anticipate it or anything else like that. Now, when we deal with the inner ear, the bony labyrinth protects the membranous labyrinth and its receptors. What are the differences? What are these things? The bony labyrinth is a shell of dense bone that surrounds and protects the membranous labyrinth. It consists of three parts: the semicircular canals, the vestibule, and the cochlea. Perilymph, which is a fluid, flows between the bony and the membranous labyrinths. The membranous labyrinth consists of semicircular canals, the utricle, saccule, and cochlear duct. So what's inside these structures? And they are filled with endolymph. Now, endolymph and perilymph cannot be mixed. If this happens, you tend to end up losing the function of the structures in the endolymph. And that's due to the ionic concentrations and I believe the osmolarity of the fluids, how they differ. The vestibule consists of the utricle, saccule, and there is a structure called the macula, which is the sensory receptors. They detect linear movements and respond to gravity. There are three semicircular tubes connected to posterior wall of the vestibule. Now, to help you, there are three on each side of your head. They are going to basically tell you information to provide you with basically what would be in the X, Y, and Z plane. So in other words, what I mean by that is sort of, horizontal, vertical, and sideways, if you want to say. On one end of the canal, there is a structure called the ampulla. It's an enlarged region that has a structure called the crista ampullaris. Inside of this structure, there are sensory receptors that detect changes in angular acceleration or deceleration. Here we have the situation of the bony labyrinth, perilymph in here, the membranous labyrinth here, and then endolymph. So wherever you see the blue, that's the membranous, and we have the semicircular ducts. If you notice, they basically are extending out in basically the three orientations of space. Now you're going to have these two other structures here, the saccule and the utricle. Fluid is going to go through them and move through them. The sensors are where you see these ending sites here, 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 as well as over here and here. Okay? Now, this is the cochlea. Notice it looks like a big snail shell. It's not big, that's for sure. And we have the vestibule in this area here. And what you're going to note is this. The cochlear duct is the processing of hearing. We're going to get into this in a few minutes. The vestibular complex, the semicircular ducts are here, here, and here. The utricle and saccule are right here. That's the membranous labyrinth in a nutshell. Now what happens? You have hair cells in these semicircular ducts, and they are basically respond to rotation. Hair cells in the utricle and saccule respond to gravity and linear acceleration. When I say linear, think about it in straight line, as opposed to going around and around and around. Each side of the head contains one set of these semicircular ducts, which are anterior, posterior, lateral. The ampulla expanded site in the ducts contains these receptors, and in the ampullary crest, there's a site of receptors that's bound to the ampullary cupola. This cupola is a gelatinous structure with hair cells embedded into it. So as the cupola gets moved around by the fluid direction, it's going to basically yank on the stereocilia or the kinetocilia. And this push or pull of the ampulla crest is going to trigger the hair cells. Now there is a difference though, and we're going to show you this in a diagram in a second. At the utricle and saccule, they have what is called an otolithic membrane. Now what I mean by that is this: not just you having some gelatinous structure and in it are the hair cell hairs embedded into it, but to provide mass and allow a greater sensitivity for linear acceleration, this otolithic membrane is embedded with calcium carbonate crystals. They're called otoliths. Now, in the macula, the hair cell dense sites are embedded in the otolithic membrane. The push or pull of the hair cells depends on the direction of the linear force. The utricle is sensitive to changes in horizontal movement. So think about it this way. You drive forward, you stop dead. You drive forward, you stop dead. You're in traffic. The saccule is sensitive to changes in vertical movement. Think about if you were going up and down on an elevator and you go up to a certain point and it dead stops for a second. Whoops, that's a scary feeling. Or it drops. That's a scarier feeling. So let's go over this. Here we have the semicircular canals, and at the end of them is this enlargement, the ampulla. And here you have the ampullary cupola. Notice that there's fluid on each side. The hair cells are here, and they have embedded structures. When the fluid is moving in this direction, it's going to yank on the cupola and thereby yank on the hair cells. Underneath the supporting cells and underneath the hair cells is the sensory nerve, which will be fired off. Now, notice you have this. If the direction of the endolip movement is relative to the direction of the duct rotation, so in other words, you put pressure on this direction, it tilts this way, you have a certain firing pattern here. You do it in the opposite, it has a different firing pattern. And if you notice, you have it in the X, Y, and Z positions. All of this in tiny little structures embedded in the inner ear. But what about the utricle and the saccule, etc.? Well, we have these hair cells again, kinocilium. You have the macula. These are the cells right here. They're all nice and dense, all bunched up together, surrounded by this nice gelatinous material, and then capped on top of them to provide further mass is otoliths. So when fluid is moving or momentum is occurring, these are going to move in one way or the other. They eventually are going to yank on the hairs, which will act on the cells to cause the sensory neurons to fire action potentials. Gravity goes in one direction, gravity goes in another direction, you're going uphill, downhill, etc. And that will alter the receptors' input and output. I shouldn't say as much input, but basically output. Now that we've gone through the equilibrium and the hearing, we're going to spend some time with the cochlear. Now, the cochlear duct contains hair cells in the spiral organ, the cochlea, and that's basically the function for hearing. The organ of Corti, otherwise known as the cochlear duct, lies between chambers that are both filled with perilymph. The chambers are the scala vestibuli, the scala tympani. The scala vestibuli, otherwise known as vestibular duct. The base is the oval window. The vestibular membrane separates the cochlear duct from the scala vestibuli. It begins at the oval window and ends at the apex of the cochlea. Remember, the cochlea is going to be in a spiral shape moving upward. The scala tympani, the tympanic duct. The base is the round window. The basilar membrane separates the cochlear duct from the scala tympani. And it begins at the apex of the cochlea and ends at the round window. You're going to have a membrane that separates each of these structures from the organ of Corti. The organ of Corti you're going to have is your middle chamber. It's filled with endolymph. It is a spiral organ, and it's located in this duct, and it's part of the membranous labyrinth. This organ of Corti consists of a tectoral membrane attached to the inner wall of the cochlear duct. Hair cells. These are arranged in a series of longitudinal rows. The cells are embedded in the basilar membrane. The cells are in contact with sensory neurons. The cells also lack kinocilia, but they have sterocilia, the hairs, and they're embedded into the tectoral membrane. So the hair cells have their sterocilia, and it's embedded into this kind of inflexible membrane that's attached to the inner wall of the cochlear duct. So how do we get these things moving and so they can fire off and give their information? You have pressure waves in the paralymph that's triggered by sound waves arriving from the tympanic membrane. Now what the pressure waves are going to do is they're going to cause the basilar membrane to move up and down. Remember, the basilar membrane is also going to be sort of the floor underneath where the hair cells are located. This is going to cause the sterocilia, which aren't going to normally be able to move around. They're going to get yanked up and down, up and down, up and down, because they've got an inflexible structure above them, the tectoral membrane. Okay? And as they're yanked up and down, they basically are going to be pulling and stimulating and sending off signals to the sensory neuron. Notice that this spirals upward. Notice that you see here the vestibular membrane. You see where the spiral organ, otherwise known as the organ of Corti, is here. You can see it very clearly. That's nice, that's important, but we're going to show you an image in a second here, because what's really important is to see where this is up close. What you see is really the organ here, and in a few other places you can see it. The areas above and below it, the scalar tympani, scalar vestibuli. What's really in the middle here? That's the cochlear duct. That's where the organ Corti is. So we now magnify further. Okay? Scalar vestibuli, scalar tympani. This is the basilar membrane. This is going to basically yank up and down as the vibrations from the fluid pass by it. When the vibrations do this, this floor is going to go up, down, up, down, up, down. The outer hair cells are here. They're embedded to this tectoral membrane. This isn't going to go anywhere. So as the floor goes down and goes up, you're yanking on these hairs, and that's going to cause these cells to fire, which will eventually lead to the neurons firing off action potentials down the nerve fibers. Remember that we had a branching off of the vestibular cochlear nerve to the cochlear nerve and the vestibular nerve. Well, we've dealt with the vestibular nerve. Now we're dealing with the cochlear nerve. Here's up close and personal. Here we are. Basilar membrane here, so this would be underneath. The fluid waves move through here, cause vibrations. This is going to move up and down, which means that this whole floor here is going to be pulled up, down, and as it is, these cilia can't go anywhere. They're embedded in the tectoral membrane, so they're going to get yanked. This is going to send the generator potentials, which will eventually cause these neurons to fire. So if that's so, how does that work? Well, this is partly where we talked about transduction before, but first you have to think about this. Sound is basically waves of pressure conducted through a medium, air or water. And when I talk about a wave, I talk about areas of crowding of air molecules and areas with very, very few air molecules, sort of like waves. And that's really what you have. You have high density, low density, high density, low density, high density, low density. When we talk about wavelength, this is the distance between the two adjacent wave peaks. So you're going to have some sound waves that have a very, very narrow wavelength, others that are going to have very wide wavelengths. But then the other thing we have to talk about is the number of waves that pass a fixed reference point. In this case, we would talk about number of waves per second. That's where we get into hertz again, okay? And then when we talk about amplitude, that's the amount of energy carried by each wave, so that could be much greater or much softer. The cochlear processes sounds by hertz, and the membrane in the organ of Corti vibrates at different sites on the membrane, depending on the intensity and on the frequency. Here we have the tuning for air molecules: dense, low density, dense, low density, dense, low density. This can be converted into waves: high density, low density, high density, low density. Difference between the two peaks is what is referred to as wavelength. Amplitude is the intensity, so that would be height. When you look at the intensity of representative sounds, we can hear between what is referred to as 20 hertz to 20,000 hertz. But how do we talk about the intensity or the force of these waves? That's where we get into the concept that is more logarithmic in its mathematical measurement. It's called decibels. Quiet library or soft whispers, 30 decibels. The lowest audible sound is about 0. When you get into busy traffic, that's about 70. Now, as I start moving up, 80, 90, that's when we start getting into the dangerous time exposures. As we have found over the decades in work and other places, the greater the intensity, the shorter the amount of time that's allowed. And what happens is it goes from 8 to 4 hours to 2 hours to 1 hour to 30 minutes, etc., before you start exhibiting damage. Now, also there is a situation called the threshold of pain, and roughly it's about 140. Other documents will say 130 decibels. At that point, you're not really hearing something, you're just having pain in the ears. Okay? Now, when you look at the cochlea, if you could take that snail shell and that tube and just extend it out for a certain distance, you will find that all along that tube, all along that distance, you have variations of sensitivity for the frequency and intensity. The higher frequencies are closer to the actual start of the cochlea. The lower frequencies are farther down. And you can see this. If I talk about 16,000 hertz, it's closer to the stapes, the open window. The basilar membrane is right here. And as you have vibrations going up and down, you are going to have a corresponding bend in the basilar membrane, which means that you will activate the organ of Corti's right here of these particular cells. Notice that as you go farther and farther down, the sensitivity, or I should say the responsiveness, the resonance of the waves correlates to the distance. So farther away, lower on the hertz band. And right down here would be something around what you might call 20 hertz. Now, I'm just trying to give you some basic understanding of this. So basically what happens is, as the waves pass down, they travel all the way down and all the way back. And what happens is, these waves will cause the basilar membrane that is basically against that organ of Corti section. It will move downward here, corresponding to the specific frequency of those waves. Okay? Now, when we talk about the vestibular pathway, we would say, well, look, vestibular cochlear nerves carry the equilibrium and hearing sensations to the brainstem. There's got to be a specific pathway for the vestibular sensations as well as to the cochlear. Yes, that's true. You're more usually aware of the cochlear, because you hear sounds, than you are for the vestibule, unless you have some imbalance problem. But let's start off with it. The sensations occur in the vestibule, carried down the vesicular nerve, fuses together for CN number 8, the vestibulocochlear nerve, passes up to the vestibular nuclei, which are in the medulla oblongata. Information is then passed to the cerebral cortex, the cerebellum, and the motor nuclei in the CNS. The cochlear pathway goes from cochlea to the cochlear nerve, to CN number 8, again the vestibulocochlear nerve, to the cochlear nuclei, which are located in the medulla oblongata. The information ascends to the superior ovulatory nuclei of the pons and both inferior caliculi of the midbrain. The pathway of sound information then passes to the medial geniculate body of the thalamus. This is where we're getting our final instructions before we send it over to the temporal lobe. Okay, so the thalamus has projection fibers that pass the information to the auditory cortex of the temporal lobe for further processing. As you can see here, this is the process of the vestibular information going eventually to the structure here, which is the vestibular nuclei of the medulla oblongata, and then it's passed upward as well as in some cases downward to carry instructions to peripheral muscles, etc., reflexive movements of the head and neck. Now, the pathway for the cochlear starts at cochlea, cochlear nerve, vestibulocochlear nerve 8, then it goes over to the basically the cochlear nuclei of the medulla oblongata, the superior ovulatory nucleus, to the inferior cochleus caliculi, then over to the medial geniculate body of the thalamus, and then over with projection fibers to deliver this information to the auditory cortex of the temporal lobe. Now that we've completed all of this, I bet you're thrilled. Maybe not, maybe so. I hope so. Anyways, this last chapter, you were to peruse chapter review outline on pages 587 to 591. Complete review questions on pages 591 to 592. Complete the review sheets on pages 552, 570, and 586. The next step is to prepare for the lecture exam number 3 and comprehensive final exam. Thank you very much for your attention.